So far we have seen how the cell cycle gets regulated. Now we are about to study how the cell cycle regulators gets controlled or regulated. So there are different mechanisms. The first one is subcellular localization. Other one is CDK inhibitors. Third one is the balance between different regulators, and another mechanism is by means of proteolysis, a controlled proteolysis. This is mostly by means of a proteasome. So coming back to the first one, that is subcellular localization. For example, in the case of cyclin B, they may be transported from the nucleoplasm into the cytoplasm during most of the times. That means during most of the times, a large amount of cyclin B that may be present in the cell cytoplasm, uh, and in that cytoplasm they will be remained as inactive. This is because of a kind of a phosphorylation, that means a kind of inactivation that may be carried out by V1 proteins that we have already studied. That means in that cytoplasm, the cyclin B remained as inactive due to a kind of inactive phosphorylation. But just prior to this onset of mitosis, this cyclin B gets localized into the nucleoplasm or into the nucleus, and then their inhibitory phosphorylation gets removed, and then the cyclin along with the CDK join together to form maturation promotion factor, and that maturation promotion factor carries out the cell cycle from G2 to M phase. The subcellular localization of cyclin B that plays a crucial role in this cell cycle regulation. These kinds of localizations are possible due to the presence of nuclear localization signals or sequences into these kinds of proteins. That means in the case of cyclin B, it do have a nuclear localization sequence or a signal in that. Uh, so just after onset of mitosis, the cyclin B, those are present in the nucleoplasm. The nucleus localization sequences gets phosphorylated, and into that phosphorylated nuclear localization sequence, exporting proteins, those are getting attached. Then this exporting protein that exports the cyclin B through the nuclear pore complex into the cytoplasm. So now the cyclin B are localized into the cytoplasm. They are not at all available in the nucleoplasm. So this cell can drive the cell cycle from M phase into the next G1 phase. And in the cytoplasm, these nuclear localization sequences gets dephosphorylated, and the exportin gets rid of nuclear localization sequences, and then just prior to the next set of mitosis, these nuclear localization sequences of cyclin B gets phosphorylated in a different position, and then the importing proteins gets bind with to the nuclear localization sequence. Then the importing protein recruits this cyclin B into the nucleoplasm, and then there these cyclin B along with the CDK that can drive the cell cycle from G2 to M phase. So here the subcellular localization of cyclin B that plays a crucial role in the cell cycle drives. In this schematic representation, you will get an idea of different cyclin levels in the nucleoplasm during different cell cycle phases. See, this is G1 S phase, G2, and this is the M phase, prometaphase, metaphase, anaphase, and telophase. And coming back to the G1 phase of the next cell cycle, we have already studied that the cyclin B are recruited into the nuclear plasm just prior to onset of mitosis. So, in during G1, S phase, etc., their nuclear levels of this cyclin B are much lower, and just onset of mitosis, and in the G2 phase, their levels are getting elevated, and that causes the drive of cell cycle from G2 to M phase. And after the M phase, their level drastically gets diminished. This is by means of different mechanism, maybe by means of a proteolytic cleavage or by means of the localization into the cytoplasm that we have studied. And coming back to the next cyclin, that is cyclin A, they are mainly involved in the cell cycle drive from G1 to S phase. So their levels are get elevated just prior to onset of S phase. So here you can see its level gets elevated, and that causes the drive of the cell cycle from G1 to S phase. Here CDK are regulated by means of inhibitors. Other factors those influences the CDK are the levels of cyclins, and the second thing is the phosphorylation, CDK phosphorylations. Phosphorylation may be inhibitory phosphorylation or excitatory phosphorylations. Inhibitory phosphorylation that inhibits the kinase activities of CDK, and excitatory phosphorylation that activates the kinase activities of CDK. And another way of cell cycle regulation is by means of keeping a balance between V1 and the CDC25 that we have already studied. So here in this, the mitotic cyclins that activates the CDC2, this CDC2 that drives the cell cycle from this G2 to M phase of the cell cycle. But the thing is that the CDC2 gets regulated by means of different mechanisms. See here, this CDC2 gets inactivated by a protein that may be called as a V1. That V1 adds an inhibitory phosphor at tyrosine 15th position or at the 14th residue, that is a threonine of the CDC2. So these are inhibitory phosphates. So this phosphorylation that makes the CDC2 inactive. So the cell cycle may stuck at G2 phase of the cell cycle. Uh, we have studied that by that time the cell in volume gets increased. Once the maximum volume gets achieved, then other protein that may be known as CDC25, that CDC25 removes these inhibitory phosphates from CDC2. So this CDC2 gets free now. By that time, this mitotic cyclin activates the CDC2, and then the CDC2 now it can capable to drive the cell cycle from G2 to M phase. So by keeping the levels of this V1 and the CDC25, uh, it can regulate the cell cycle here. And another way of cell cycle regulation is by means of proteolysis, a controlled proteolysis. This is actually carried out by proteasomes in a different pathway. That pathway is known as ubiquitin proteasome pathway. Here, see, this is the picture of a proteasome. It's having four rings, of which two of them are alpha. See, this is the one alpha, and this is another alpha, and these are the beta rings. So it's having two alpha rings and two beta rings. The alpha rings are present at the outer side of the proteasomes, so that it can maintain the gate of the proteasome. Through that gate, the proteins to be degraded get into the proteasome, and beta rings having 3 to 7 proteolytic uh, sites in that. So this beta rings carries out the proteolysis. Ubiquitin ligase is an enzyme that adds ubiquitin into the proteins those need to be degraded. So the proteins those are to be degraded or destroyed are marked first by means of ubiquitin. So this ubiquitin protein complex, only these complexes get into the proteasome, and finally the beta rings of the proteasome that degrades the proteins those are marked. Here in the case of a cell cycle, different cyclins at a different phase of the cell cycle are the targets of the proteasomes, so that the levels of different cyclins, CDK, different proteins are get regulated, or their levels are maintained.